Assalamu alaikum adab. Welcome to the presentation on the topic of mechanism of urine formation. First of all, introduction. Introduction is given here in 7 points. The urinary system that is responsible for the production of urine, it consists of 2 kidneys, 2 ureters, 1 urinary bladder and 1 urethra in human beings. Kidneys are the primary means of excretion. The excretory product, that is urine, it consists of mainly urea, that is formed by the catabolism of amino acids. Creatinine comes from muscles, uric acid comes from nucleic acids, bilirubin found from the breakdown of hemoglobin. Metabolites of various hormones and the substances like the pesticides, drugs, food additives, and the ions excreted. This is the urine that is excreted from kidneys. Point number 3, the process of urine formation occurs in nephrons. Nephron is the basic structural and functional unit of kidney. Point number 4, each human kidney, it contains more than lakh nephrons, each of which is capable of forming urine. There are 3 general processes responsible for the formation of urine, that is filtration, reabsorption and secretion. Depending upon the water requirement of body either produces dilute urine or concentrated urine. The slide shows the nephron and its blood supply. Here you can see the nephron. First, it consists of this Bowman's capsule. Bowman's capsule is followed by the proximal convoluted tubule, that is BCT. It then leads into loop of Henle. This loop of Henle then is followed by distal convoluted tubule, that is DCT, and DCT then leads into cortical collecting duct and finally the main collecting duct. The arcuate artery here you see in the form of afferent arteriole supplies this Bowman's capsule, and the afferent arteriole breaks down into the glomerular capillary network, and these glomerular capillaries later on unite to form efferent arterioles. Then this efferent arteriole forms peritubular capillary network around the tubules of the nephron. The peritubular capillaries here in this portion surrounding this loop of Henle, it is called vasa recta. Here you see the slide shows the basic process of urine formation. The 3 sub-processes responsible for the formation of urine, that is filtration. Filtration happens here in glomerular capillaries in Bowman's capsule. It is followed by then reabsorption and secretion. Reabsorption and secretion occurs in the tubules of the nephron, and finally the urinary excretion. These 2 and 3, this is reabsorption and secretion, peritubular capillary network surrounding the tubules of the nephron, they play their role, and finally the urinary excretion happens. Here glomerular filtration in this slide. Glomerular filtration is the first step in urine formation. Each day about 180 liters of plasma fluid is filtered. Plasma, that is blood, is filtered in the glomerular capillaries, out of which all is reabsorbed except leaving behind 1 liter of fluid to be excreted each day. Fluid is forced through the filtration membrane. Glomerular capillaries and surrounding podocytes form a leaky barrier facilitating filtration. This capillary network, capillaries and surrounded by podocytes, they form a leaky barrier. These fenestrations or pores here are responsible for the formation of that leaky barrier, and this facilitates filtration as shown in the figure. And all the blood constituents except the plasma proteins, blood platelets and blood cells, that is RBCs and WBCs, they remain behind and all other constituents are filtered. Volume of fluid filtered here is much larger than other capillaries. volume of fluid filtered here in glomerular capillaries. The filtration is much larger than other capillaries of the body, because glomerular capillaries present a large surface area for filtration because they are long and extensive. Second, the filtration membrane is 50 times more leakier than other capillaries because of these fenestrations, these podocytes. And the glomerular capillary blood pressure is high due to smaller diameter of efferent arteriole, and the larger diameter afferent arteriole. This is larger in diameter, this is smaller in diameter, creating the pressure here. Also, the glomerular filtration depends upon the net filtration pressure. The three different types of pressures are responsible. There is glomerular hydrostatic pressure, capsular hydrostatic pressure, and blood colloid osmotic pressure. Capsular hydrostatic pressure and blood colloid osmotic pressure, they acts against this glomerular hydrostatic pressure, and the values here show the 55 mmHg, 15 mmHg, 30 mmHg. Finally, the 10 mmHg net filtration pressure, it is responsible for the filtration at the site of glomerular capillaries or Bowman's capsule. Here the slide shows renal tubular reabsorption and tubular secretion. As already told that this is the Bowman's capsule, it is followed by PCT, loop of Henle, DCT, and collecting duct. These different portions of nephrons shows differences in terms of reabsorption, secretion. We can see in proximal convoluted tubule. This is the portion where the filtration happens. It is followed by proximal convoluted tubule. Maximum absorption of substances happens in this proximal convoluted tubule. Here you can see water, 65% of water is reabsorbed through osmosis. Sodium ion, 65% through the sodium potassium pumps or sympoters and depoters. Potassium, you can see 65% is reabsorbed here through diffusion. Glucose above almost 100%. Amino acids also 100% reabsorbed in here in this portion. Chlorine ion about 50% through diffusion is reabsorbed. Bicarbonate 80 to 90% through facilitated diffusion is reabsorbed. Urea about 50% is diffuses back into the blood. Calcium and magnesium, they also diffuse at variable amounts. Secretion also happens here. Not only reabsorption, but secretion also happens. Secretion of this H positive ion, ammonium ion, urea, creatinine. At end of PCT, tubular fluid is still isotonic to blood, and the tonicity about 300 milliosmoles per liter. Now you can see in nephron loop. This is the nephron loop. It is the thin portion and it is the thick portion of this nephron loop. It is the descending portion, it is the ascending portion. Reabsorption into the blood, we can see water about 15%, 65% has happened here, and now 15% here in this portion only, because this is impermeable to water. 15% is diffused through osmosis. Sodium 20 to 30% here, potassium 20 to 30%, chlorine 35%, bicarbonate 10 to 20% through facilitated diffusion. Calcium and magnesium also in variable amounts, they diffuse. Secretion also happens here in this portion, that is this in this loop. Urea, it is important to mention here that this is the portion which is permeable to urea. This is impermeable. This is impermeable to urea. Urea recycling happens here. We will be seeing later on how this urea recycling happens. At the end of nephron loop, tubular fluid is hypotonic, that is 100 to 150 milliosmoles per liter. That is when the fluid reaches here, when it ascends up in the ascending limb, it turns hypotonic. Now this early part of this distal convoluted tubule, that is DCT, reabsorption happens water 10 to 15% through osmosis here, 10 to 15%. Sodium 5%, chlorine 5%, and calcium variable. It is stimulated by parathyroid hormone, the reabsorption of calcium here. Late distal convoluted tubule and collecting duct. These are late. This is from here. Late distal convoluted tubule and collecting duct. You can see water 5 to 9%. It is under the effect of ADH, anti-diuretic hormone. Sodium 1 to 4%. Bicarbonate variable amounts, and then urea, it is recycled. Urea recycling will be seen later on how it happens. Secretion into urine happens, potassium and this hydrogen. Adjust this variable amount to adjust for dietary intake. Leakage channels, potassium leakage channels are here, and they led to the leakage of potassium through secretion. Similarly, this H positive ion, variable amounts to maintain acid base balance, that is hemostasis. H positive pumps are here to maintain the acid base balance. Extra H positive is being excreted out. Tubular fluid leaving the collecting duct is dilute when ADH level is low and concentrated when ADH level is high. This distal convoluted tubule, importantly the last portion of the distal convoluted tubule, and the distal convoluted tubule, it works under the effect of ADH. When ADH is secreted, the principal cells here turn permeable to allow water exchange, to actually allow water reabsorption here. And when ADH level is low, it is impermeable to water and does not allow the water to get reabsorbed here in this portion. So we see maximum reabsorption happens here. This portion you see, this is permeable to water. This is permeable to water. This portion is impermeable to water, but symporters of ions are present there, and they symport the ions out into the this medullary portion, and this very portion, that is distal convoluted tubule, or the upper portions of this collecting duct, they work under the conditions of ADH. If ADH is present, this is permeable to water. If ADH is absent, this is impermeable to water. Now the formation of dilute and concentrated urines. As already told, that this descending portion of this descending portion is permeable to water. Descending portion. Here you see this very deep dark brown. It is impermeable to water, but it allows the symport of ions out. This blue portion, it is impermeable to water when ADH is absent. When ADH is secreted, in the presence of ADH, it turns permeable to water. Now, how the dilute urine is formed? Actually, when the fluid content inside the tubule, it descends down, water moves out. Water moves out under the effect of this increasing osmotic gradient here. How this osmotic gradient is formed? Osmotic gradient is actually found by the symport of ions Na and Cl into the this interstitial fluid of medulla. When the fluid content descends down, water is reabsorbed here, and in this portion ions come out and it is impermeable to water. And now here if ADH is present, the water is reabsorbed. If ADH is not secreted, water is not reabsorbed. And when there is enough water inside the body and no ADH is secretion, at that time the urine excreted is actually hypotonic, and the tonicity gets on decreasing as it ascends. Continuously, because ions come out and tonicity decreases, it turns hypotonic. And when no water is further reabsorbed, and the dilute urine is actually excreted, and the dilute urine is about 4 times more dilute than this tonicity, that is 300 milliosmoles per liter. The filtrate, when it enters PCT, it is 300 milliosmoles per liter, but when the urine leaves, it is about 4 times more dilute than this 300 you can see here. And the effect of the ADH, it plays its role for the formation of dilute urine. Now the formation of concentrated urine. Highly concentrated urine, which being about 4 times more concentrated than plasma, is produced by kidneys under high water scarcity conditions in body. ADH is responsible for it. The ability of ADH to cause excretion of concentrated urine depends upon the presence of an osmotic gradient, depends upon the presence of the osmotic gradient of solute in the interstitial fluid of renal medulla. The osmotic gradient in turn depends upon two main factors: difference in solute and water permeability of the nephron. As already told that this is permeable to water, this descending portion. Ascending portion is impermeable to water, but symports of ions happens here in this portion. The permeability of water of this late DCT and collecting duct, it depends upon the presence or absence of this ADH. This difference in solute and water permeability of the nephron, it is responsible, and another factor that is responsible, it is counter current mechanism. Counter current mechanism. What is the counter current mechanism? It is that in one tube it is flowing down, and another tube it is flowing up. It is flowing in opposite directions in the two parallel tubes, and this is counter current mechanism. Now, how the concentrated urine is produced? It depends upon four factors. One, the symport of ions in thick ascending limb. Thick ascending limb reabsorbs sodium, potassium to chlorine symport and causing buildup of sodium and chlorine ions in interstitial fluid of the medulla here. Second is counter current flow in the ascending and descending limb. It establishes the osmotic gradient in the renal medulla. The symport in the thick ascending limb continuously pump out ions in the interstitial fluid. It continuously pumps ions in the interstitial fluid. The tubular fluid loses water as it descends down here. Loses water as it descends down, thus becoming more concentrated. And when reaches ascending and when creating osmolarity of about 1200 milliosmoles per liter at the hairpin turn of the loop. When this fluid reaches here at this point, the concentration increases so much that it is around 1200 milliosmoles per liter. The third point is the cells in the collecting duct absorb more water and urea in the presence of ADH. In the presence of ADH, more water is reabsorbed. This is the portion which is impermeable to urea. You can see these portions are permeable to urea. These portions are permeable to urea. This is impermeable to urea. Okay, this very portion. As the urea concentration, because increased in concentration due to water loss, urea is also diffused inside here. This increases the concentration further, and here the movement of ions out, urea is not excreted here. And when under water scarcity conditions, water is reabsorbed here under the effect of ADH, the concentration increases further, and when it reaches here, urea, due to increased concentration, urea diffuses out, or it is reabsorbed and it reaches here in this portion. In this way, urea recycling happens. Urea comes out here and it diffuses into the loop of Henle portion here. This leads to the urea recycling. In this way, reabsorption of water from the tubular fluid of ducts promotes buildup of urea in the interstitial fluid of renal medulla, which in turn promotes water reabsorption. And water reabsorption promoted, it increases the tonicity of the urine, and hence highly concentrated urine is produced. This counter current mechanism, it two sub mechanisms, that is counter current multiplication and counter current exchange happens here. What is this counter current multiplication? It is the counter current flow here in the loop of Henle, it increases the osmotic gradient. It is the countercurrent multiplication. Another is the countercurrent exchange. Countercurrent exchange, you can see here. Countercurrent exchange, actually it is the vasa recta portion, the peritubular capillary network surrounding this loop of Henle. It is called vasa recta. Vasa recta has also been shown here. Vasa recta also consists of a descending limb and ascending limb. Countercurrent flow also happens here. In one limb you see it is descending and the flow is descending, and in the opposite you can see it shows ascending. The vasa recta also has ascending and descending limb, as already told. This countercurrent flow uh, in this countercurrent flow, you see here in this portion, as the fluid inside the vasa recta descends down, the blood inside the vasa recta descends down due to the increased osmotic gradient. Water diffuses out and these salts diffuse in, and reaching down, you see the process continues, water out and NaCl inside, these ions inside. And when the blood inside ascends, you see here now, due to the decrease in osmotic gradient upwards, water diffuses in and ions diffuse out, ions come out. And now the vasa recta is playing the role of here the countercurrent exchanger, actually, because water comes out here, here water goes inside. It means that no net change of osmotic gradient happens here. It is only the maintenance of osmotic gradient and the role that is being played by this vasa recta. The countercurrent flow here, it increases the osmotic gradient. The countercurrent flow here, it maintains the osmotic gradient. Countercurrent flow in vasa recta, it maintains the osmotic gradient, whereas it increases the osmotic gradient. And this osmotic gradient increase, multiplication or countercurrent exchange, as well as the factor of ADH, all these factors collectively are responsible for the formation of concentrated urine, and concentrated urine is about 4 times more concentrated than this 300 milliosmoles per liter fluid that enters into the PCT. These are the references. Thanks.